And today I will delve into my doctoral research, which is a feasibility study entitled Targeted Cadmium Species Analysis in Blood for Occupational Exposure Assessment. And this work is conducted in collaboration with the Institute of Science and Physicochemistry for Environments and Materials. And during this presentation, so I will briefly explain the origin and the objectives of the study, and then the method which employed to obtain the results, and finally the future work. And so let's begin. As you know, probably, cadmium is a toxic element which is carcinogenic, and it can promote kidney failure, liver diseases, osteoporosis, and despite rich regulation, we can find cadmium in several industry sector and uses. So it's overall about 22,000 workers are concerned in France by cadmium exposure. And to evaluate biologically workers exposed to cadmium, we can use these guideline values in the biological matrix. And we can just say that urine is the preferred matrix to evaluate an exposition of cadmium, as it's very easy to sample and it reflects more a long-term exposure than blood. So here are just some additional values in the airborne, but the most important thing to know here is that a low cadmium exposure can lead in a urinary cadmium, which is in close proximity to the renal critical dose effect. So, concretely, we conducted a field study in the battery recycling sectors, and here the only thing I would like to say is that we observed some very high value, but very punctual, and this value conducted to very punctual value in a very punctual high value in the urine. So this high value in urine were more a kidney saturation effect than a real evaluation of cadmium in the urine. So, but this prompts several questions, such as how to be more efficient and proactive to improve this risk assessment, because as you probably know, cadmium is an element which is stored in the kidneys, and if there is a kidney saturation effect, workers are potentially exposed to this element. So from this question, we can this leads to my objective. But before expose my objectives of the study, let's have a look on the toxicokinetic of cadmium. And we can say that after an ingestion or inhalation, cadmium enters rapidly into the bloodstream, and then it is transported to organ, and it can complex to different proteins such as albumin, metabetonin. It is stored in the red blood cell too. But here, and from this toxicokinetic, we decided to focus on blood biomarkers, as we can find in the bloodstream a lot of different cadmium species. And indeed, the main objective of my work will be the determination of cadmium species, as it the main species we can find in the bloodstream inform on the kind of exposure, so short-term exposure or long-term exposure for metallothionein complex, for example. And so my objective is divided into two axes, which is development of analytical methods and an application to real sample in a field study where workers are mainly exposed to cadmium. And yes, how can we do this? So to analyze the different cadmium species, we need a blood fractionation and a specific analysis in the plasma and a specific analysis in red blood cell. But today I won't speak about the plasma fraction, as it's still a work in progress. But let's have a look on the red blood cell fraction, and we decided to analyze red blood cell by a single cell, and the quantification by inductively coupled plasma and with a triple quadruple mass spectrometry. And it consists in the analysis of individual cell and quantify the elemental concentration in each individual cells. And we opted to a triple quadruple ICP-MS, as it is very sensitive, and thanks to an efficiently management of interferences. So yes. As you can imagine here, the main difficulties is the injection of individual cells in the ICP-MS, and for that we established a sample preparation, which consists in the isolation of the cells and a fixation of the membrane of the cells with glutaraldehyde. And with such protocol, we succeed to prepare a cell suspension in water, and we perform a 10-fold dilution in water. And the most interesting is that these cells are stable for up to 90 days. And then after this work, we decided to validate the quantification of the elemental concentration in the cells. And for that, we established a strategy for the method validation. And we compare our new method to a reference method, which consists in a mineralization procedure and an analysis of large samples with an ICP-MS. And unfortunately, we were obliged to use blood from a general population, as it is blood from donors from the French establishment of blood. And unfortunately, they do not contain cadmium. However, they do contain other elements which are native in the red blood cell. And we decided to focus on this element to validate the method of quantification and then extrapolate the parameters of the method to other elements such as cadmium. So, okay, just an example of some signals obtained from an analysis, a single cell analysis for this element, and the only thing here is that we can see that injection and detection were adequate for our work. And then let's have a look at the accuracy and sensitivities. The first column concerns the limit of detection in attogram per cell in for the single cell analysis. The second column concerns the elemental concentration in the cells, and the third column concerns the analysis by bulk analysis and bulk ICP-MS analysis, so our reference method. And this value was corrected by a cell counting with a microscopy, by microscopy. And we can see that for this element we had adequate accuracies and sensitivities compared to our reference method. So for blood from general population, this method seems to be adequate. For these two elements, we were not able to analyze for both bulk ICP and MS and single cell analysis. So this was not adequate for the quantification of cell in the general population. And for these elements, we had an adequate quantification for our reference method, but not for the single cell analysis. So we can imagine that single cell analysis is perhaps not enough sensitive for this element, but just we can look that for the lead we had limit of detection which were in close proximity to the reference method. So the question is now that it needs further validation and further analysis from samples who are exposed from exposed workers, but we know that our quantification methods work on elements which are abundant in the cells. But we found that single cell analysis have many advantages, and the most important is that between 1 to the 4th and 1 to the 5th cells are needed, which represent less than 0.1 microliter of whole blood. So an article has just been submitted in Talenta Journal for this part. And for the future work, I will focus on the plasma fraction, and then I will study. Yes, I will apply these techniques to a field study. Thank you.
Okay, thank you, Mathieu.